I'd like to introduce Marco, who is actually from UCLA, not the Children's Tumor Foundation. But he did, in fact, fly his own plane up here, from what I hear. And he works on NF2 and is going to give us a clinical perspective on NF2. And oh, by the way, I have also been asked to announce, if you use Twitter, please use hashtag NF2Hack. Yeah? Yeah. Yeah. Hashtag NF2Hack. All right. With that.
Hello, everybody. I made it in time. Well, I'm Marco Giovannini. I'm a professor of head and neck surgery at UCLA. And I've been working on NF2 research for the last 20 years at least. And I had the chance and honor to be part of the team which identified the NF2 gene back in 1993. And then at the time I was a young postdoc. Then my next project was to develop mouse models of NF2. So mice where we reproduce the genetic mutations. The mouse has an NF2 gene which is almost identical to the human. So we just introduced mutations that are frequently found in patients and in tumor patients. And at the end, you know, after lots of crossing mice, etc., we were able to obtain mouse models of the two most frequent tumor types in NF2, schwannomas and meningiomas. And we have been using, we and others, we have distributed these mice to more than 150 labs around the world. And using those mice, we try to understand, on one hand, what are the functions on the NF2 Merlin protein, and on the other hand, see whether we can use these models to test new therapeutic approaches. So I was very happy a couple of years ago when our mouse predicted that one drug called RAD001, or everolimus, a derivative of rapamycin, an inhibitor of the mTOR pathway, showed a positive signal in all the mouse models we tested. And based on that, and based on observation in an index patient treated in Paris by Dr. Kalamaridis, we started 3 clinical trials, actually. And in the end, the results of the clinical trials showed that the mouse model was predictive of several outcomes, several observations we made in those patients. So that was a nice way to go back and forth from human to the mice and back. And hopefully this, you know, we will be able to use these models in future trials. So this is new for me. The reason why I'm here is that I was very impressed. I mean, from the first time I met Onno, I thought, well, this guy is something different. You know, of course, you know, I learned his personal story, but also, you know, how smart he is, and the idea of being part of a community where, you know, people would come in here thinking out of the box is extremely refreshing for me, and I wanted to be part of it. So when we spoke a week ago saying, I'm coming. But at the same time, I come with, you know, my old school traditional background. And so I don't want to influence you in any way, in the sense that I don't want to filter, you know, the creativity and the potential, you know, any potential outcome of this reunion. So I was thinking that maybe my role would be to, the best way I could contribute to this event is by giving you clues on what is the disease and some basic fundamentals of the genetics involved in this disease. So I have no idea what your background is. You probably outsmart me in many fields, but so I try, to give you an average level degree of information you might find boring, and of course we know it, or maybe you find something interesting. So let's see. So neurofibromatosis is part of a group of 3 diseases. Collectively, these are called the neurofibromatosis. So neurofibromatosis type 1, that was called von Recklinghausen disease. And what is interesting is NF1 alone is the most frequent tumor predisposition in humans, the frequency being of 1 in 3,500. The second type is NF2, so 10 times less frequent, 1 in 33,000. And the last type, NF3, or familial schwannomatosis, is a kind of a newer definition of the disease. For a long time it has been assimilated for NF2, and only genetic analysis has shown that in fact it was due to different genes, but still the NF2 gene is involved, so it's kind of a hybrid form between NF2 and something else, which makes it extremely interesting. And NF2 as an individual clinical form was defined by Wishart in Scotland in 1822. It was also called a central form of schwannomatosis because most of the tumors occur in the central nervous system, in contrast to NF1, where the periphery, the skin, and peripheral nerves are involved. So it's only in 1987 when the 3 entities were officially recognized as 3 different diseases, and that basically because of molecular studies, cytogenetic, chromosome analysis, and linkage studies that showed that the 3 genes were located on 2 different chromosomes. So chromosome 17 for NF1 and chromosome 22 for NF2 and schwannomatosis. You can interrupt at any time. This is not formal. Not a formal presentation. So about 92% of individuals with NF2 have been shown to carry NF2 mutation in the second or greater generation of family. Why is that? The reason is that about half of NF2 patients are sporadic cases, so they are new mutation. They don't have a family history. So it simply happens that the mutation occur during their embryonic development. And of course, the phenotype becomes fully penetrant in the following generation, because about 30% of that 50% of sporadic NF2 cases are mosaic, meaning not all cells in their body carry the mutation. So that gives rise to milder phenotypes, so maybe and segmental forms, patients that have only tumors in a leg or only in the vestibular nerve. And but of course, once the mutation is transmitted to the progeny, it becomes fully penetrant. What else? Yes, loss of expression of the NF2 protein, Merlin or schwannomin, has been shown in both sporadic and NF2-related schwannoma. Okay, so this is a first clue. Schwannomas are frequent also in the general population, not only in NF2 patients, not only in people who have a germline mutation of the NF2 gene. So any of us can develop a schwannoma. And together with meningiomas, which is the second more frequent tumor type in NF2, they are the most frequent tumors of the nervous system in humans. So also sporadic, again, not NF2 patient, general population people, will develop a tumor in their lifetime with an underlying sporadic NF2 mutation. So pure random chance of getting an NF2 mutation, resulting in the same tumors that we see in NF2 patients, but also in other tumor types. For example, mesothelioma, which is a malignant tumors of the pleural, the membrane around the lungs, which is usually triggered by professional exposure to asbestos, carries a high frequency of NF2 mutation. About 60% of these tumors have NF2 mutations. But also kidney tumors, breast, melanoma, liver tumors can carry NF2 mutation, usually in addition to other gene alteration. So this is important because if we find a treatment or a new therapeutic option for NF2, that option could have much broader therapeutic application, and pharmaceutical companies like that. So most
Would it be possible to use genome edition for this kind of disease?
to use what?
Genome edition, like CRISPR. Yeah,
Gene therapy. Would it be possible to use gene therapy now that you can pinpoint the genes related?
Yes, so you are referring to gene replacement therapy in this case. Science fiction, but yes. In theory, yes. If you find a way to bring the CRISPR, vector to the cells that carry the NF2 mutation, of course you must know the mutation in advance, but then you can engineer a vector. The vector goes in the cell, and the cell, the vector fixes, the mutated gene, and the protein is re-expressed, and thus everything goes back to normal. That's the concept. I don't think that anyone is there yet. But, there are other genetic diseases where this, has been achieved. For example, in a rare form of child immunodeficiency. That was a team in France when I worked there, and I know them quite well. But of course, then they had roadblocks because they fixed the immunodeficiency, but then the kid got leukemia because the virus integrated somewhere else in the genome and screwed up another pathway. Okay, so I think this gentleman was first.
Have the NF1 and NF2 genes been replicated in mice?
Well, mice have their own orthologs. So there is a mouse NF2 gene, there is a mouse NF1 gene, and they look.
The same as human sequence?
The sequence is slightly different, but the amino acid sequence is almost identical. For NF2, there is one amino acid difference.
So in that case, mutations are different between humans and mice compared to the normal.
Meaning, if you want to introduce a mutation in the genomic sequence, you can find a way. We found ways to mimic the human mutation in mice. I mean, you can approximate. Yeah. Okay. I don't know if I can hear you. But
Maybe we'll hold discussions
we hold.
until a little bit later. Yeah, just there's a
I'll
lot
be
of
around
people
all
here.
weekend, so. Unless you kick me out.
And these are great
mutate.
questions.
So, I already mentioned that mutations of both alleles of the NF2 gene have been found in tumors from NF2 patients, but also in patients with sporadic vestibular phenomena. So those patients, general population, suddenly unilateral hearing loss, there is a tumor. That's what we mean for, sporadic vestibular phenomena. So both sporadic and NF2 carry inactivation of both genes, of both alleles. So just to give you, to summarize what is genetic mechanism, this is a classic, from a genetic perspective, this is the classic mechanism of inactivation of a tumor suppressor gene. Okay, so both copies of the gene must be inactivated in order to develop the tumorigenic potential of that mutation. So one allele is not sufficient. So in the case of NF2 patients, one copy is mutated in the germline. So every cell of the body carry one NF2 gene mutation in one chromosome. And what happens, the second copy, and usually it's a point mutation, then the second allele of the NF2 gene is mutated, this time usually by deletion, or can be another point mutation, but usually it's a more or less large chromosomal deletion, resulting in biallelic inactivation of the NF2 gene. So if you look in the blood or in a fibroblast, in whatever cell of the body of an NF2 patient, you find one mutation. If you look in the tumor, you will find two independent mutations. Okay, so this is the classic tumor suppressor gene inheritance and mechanism of tumor development. In the case of the general population, so people who carry two wild-type normal NF2 alleles, both hits occur in the cell of origin of schwannoma or meningioma. So there is a normal Schwann cell. Suddenly, this normal Schwann cell loses a copy of the NF2 gene. And then, sometime later, the same cell loses the second copy, and the tumor starts. Questions on this?
Any methylation process involved?
Rare. So where methylation could be involved is in this event, right? In the second hit. So there was a study done in the UK by Dr. Evans, and they showed that in contrast to other, diseases, mutation, so inactivation, the second copy of the gene by methylation of its promoter, reducing, expression or abolishing expression, it's very rare in NF2. So
it's prevalent in X chromosomes, right? Methylation of the other.
Yeah, that's the way the X chromosome 1 is inactivated.
But on this one, it's rare.
It's rare, and this is chromosome 22, so it's not X-linked. But yeah. So I'm just repeating, using a different slide, but the concept is the same. Okay, this is an MRI scan coronal view of an NF2 patient head. This patient had the three tumor types that are more frequently associated with neurofibromatosis type 2. Bilateral vestibular schwannomas, ependymoma, about 4 to 8% of NF2 patients also develop ependymomas, a kind of central nervous, a kind of glioma, and meningioma. About 50% of NF2 patients develop meningioma. So if we do genetic analysis of these tumors, we found that all tumors carry biallelic mutation or inactivation of the NF2 gene, so they are NF2 minus. But if we test a normal cell from this patient, only one NF2 gene is mutated. Okay, so the concept of germline mutation. So this is something controversial that I want to throw out there because I think it's highly relevant to what you guys are going to do. Okay, it's clear that, and also from our experiments in the mouse, there is a number of evidences that inactivation of the NF2 gene is necessary and sufficient to initiate tumor development. Necessary meaning it has to occur. Sufficient is doesn't need other gene alteration or genome modification or epigenetic modification to result in schwannoma initiation. However, and I'm not a mathematician, this group from Canada actually, but it was like Canada, US, and UK, came out with this mathematic model suggesting that yes, the two-hit hypothesis, so two copies, but a three-hit model could also fit very well in the genetic of NF2. So this suggests that yes, you need biallelic inactivation of NF2, but you also need something else. But no one has ever continued, taking this avenue. But I thought this is highly relevant for what you guys are doing. And that third hit could be something different in every tumor, or could be something different in a family of genes that all work in a specific cell signaling pathway, for example. So that's...
We have a quick question over here.
Do we have any understanding, either structurally or genomically, why it is very common, or not very common, but it is this spontaneous deletion is happening frequently in this NF2 gene?
No. So if I understand the question is why this locus has sort of an intrinsic predisposition to mutation, right? No, but that's a general question. For the NF1 gene, it's a gigantic gene, so I guess just the chance of a mutation occurring there could explain some, but still it's too frequently mutating. Just to think about, the size. No, I mean, that's an open question, not just in NF2, for every, in tumor genetics, why some gene loci are more vulnerable and predisposed to mutation or alterations than others. Yeah, that's an open field, definitely, yes.
Could the third hit be an EGFR mutation?
I've heard of one. What is interesting about EGFR is that functional studies from the very beginning, after the gene was and the protein was identified and studied, showed that there is a link. So Merlin, the NF2 protein, is just downstream of the EGF receptor. So the EGF receptor is a transmembrane protein, and Merlin is some membrane protein. So there are studies showing that Merlin is downstream of EGF receptor. It makes sense. Oh, and there is another. Yes, that was a very old study. Very old studies, we don't do those anymore. But if when they used to do, what was it called? Chemical mutagenesis, like nitrosurea. So they gave, these agents that generate breaks and random mutation in the genome. So they used to give these drugs to mice. And many, actually rats, developed schwannomas of the trigeminal nerves. It was one of the most frequent tumors obtained using that using that strategy. So when they sequenced, or I don't know what they did, because we didn't have the power to do massive genome analysis, so I think it was probably a focused candidate gene kind of approach sequencing. They found that nitrosurea was inducing EGFR point mutations. So there is a link between EGFR mutations and schwannomagenesis. Usually when you mutate EGFR, at least in rats, the tumors started as schwannomas. Oh, that's something I have to mention. Schwannomas are benign tumors, so I will come back to this, very important. The rat tumors quickly transformed and they became MPNST, which are malignant schwannomas. So MPNST is malignant peripheral nerve sheath tumor, suggesting that that direct mutation of AGR would result in a much more aggressive proliferation than that induced by mutation of the NF2 gene. Because our hypothesis, we published this a few years ago in my lab, is that the role of Merlin is to intervenes in the recycling of receptors at the membrane. So brings and takes out receptors. So especially recycles, so takes out once they're done, they've done their job at the membrane. So in the absence of Merlin, these receptors tend to accumulate. So you have a kind of a upregulation of the EGF pathway without having that strong oncogenic dominant effect that we see in the rat. I don't know if this is clear, but.
Marco, one question. Just to maybe clarify this concept, which I think is very relevant for the audience because it can, suggest something really new. So let's say that...
Oh, this is very old, 2000.
It's very old, but I mean, we can...
Simply no one has
have
been
a
working
fresh
on it.
view on the disease. So let's say that the 3-hit model, the hypothesis would be that you still find some tissue which has 2 hits, which is not a tumor. Right?
Possible.
When you say 2 hits, both membranes are here.
I mean,
Well,
what
not necessarily
I think,
NF2 hits.
No,
It could be
okay.
2 hits in another gene, like what happens in schwannomatosis.
Okay.
Could be 2 hits in the SMARCB1 gene.
and then the
and
third
then-
one is connected to the second allele of NF2.
Yes, to the
Um,
first allele of NF2, and then the second allele of NF2 is mutated
So let's say,
somatically.
how do you actually verify this? We need to take more samples from the guy. I mean, no, but I mean,
It's from one clone, same clone of the cell.
Say again.
Genome. His complete genome sequence.
Is from the blood, so from a germline.
One specific
No.
clone of cells.
No, from a tumor biopsy.
yes. So
You'll have a mix.
But these tumors are quite homogeneous. So it's a very uniform cell population. It's not like Neurofibromatosis in NF1 when you have Schwann cells, fat cells, fibroblasts, mastocytes. Schwannomas are, and as are meningiomas, the other tumor type associated with NF2, are almost exclusively composed of Schwann cells or meningeal cells. Of course, some tumor vascular endothelial cells, here and there, but those are not mutated, so you can filter them out.
So part of my role here is to keep us on time, and I think it's
I'm almost
really,
done.
really important. I know. Some of these discussions are amazing, but what I would suggest is that Marco goes around to each of your teams, and you can have that discussion with him as the weekend goes on, and that's really going to be incredible. So, please.
What?
Or links to the slide.
Oh, this one. Well, maybe we can download the paper,
Yeah.
right?
On the
Yeah, we
Where
can pop
is
the
it?
paper up on the site. That's no big deal.
Oh, genetic epidemiology. Yeah, we should be able to
Yeah.
download it. We
should
can,
get it.
the
Yeah.
PDF, yeah. So what's going on? 2016, 2017, 2 papers came out on the genomic landscape of schwannoma. Of course, this is the fashion. You cannot, if you try to publish a paper saying, oh, I sequenced a human schwannoma, it's already not enough. So you have to say genomic landscape, meaning DNA, RNA, methylation profile, and, more, whatever, just more. So basically, this is what happened, especially the first one. I think it was a fantastic marketing success. And the second one, European, Sweden, came out later, of course, lower impact journal. Sorry, I'm just drifting. Anyway, they showed that, I mean, basically nothing new in the sense that, of course, NF2 mutations are important for schwannoma. We knew it. And then they show mutations in other genes. And if you look, I don't know if you guys have experience in tumor, in sequencing of tumors, but whatever tumor you sequence, there are ARID1 and 2 mutations. I don't know why they found ARID1 and 2 mutations. The first paper also identified a chromosomal translocation.
Is that 2217 translocation?
Oh, I don't remember.
Is that 17, 22
No. Nothing to do with,
chromosome?
no, different chromosome, different genes. I don't know. Can you please look if you see it in this tumor? The second group didn't find it. What else? Well, maybe we can, I think I gave it to you, Ono, both PDFs. I think it's important. What is important is also, since, these papers are published, these people, in theory, are supposed to distribute the data, the raw data. So I think that adds a great deal of power to any genomic analysis, just merging. Oh, one big difference is both papers were on sporadic schwannomas, not specifically on NF2 schwannomas. I threw just questions. Something that I always found extremely intriguing and intellectually challenging is why mutation in such an important gene as the NF2 gene result only in 3 tumor type. Okay, so what are the determinants of the tumor spectrum? Why the tumor spectrum in these patients is so narrow, so restricted? Why all these tumors are benign tumors? So what is a benign tumor? A benign tumor is a tumor that does not invade the neighboring tissues, does not give metastasis, and does not spontaneously, most of the time, in the case of schwannomas, they never undergo malignant transformation unless irradiated. So there is a huge debate on the use or non-use of radiosurgery for NF2. Unfortunately, this is more and more frequent, not in our clinic. And I can elaborate why we are against it. Meningiomas is a different story. 90% of meningiomas are grade 1, so benign tumors, but the other 10% are grade 2 and 3, so progressively more and more malignant, so atypical and anaplastic. Ependymomas are benign tumors, but since they grow in the spinal cord, of course, patients, eventually become paralyzed, and that's another therapeutic or clinical management challenge. But usually they respond well to surgery when it's well done. So are the mechanisms, the tumorigenic mechanisms initiated by NF2 loss tissue specific? Could be tissue specificity the key to explain that restricted tumor spectrum? That means there might be cell types where loss of NF2 has no effect. Either kills a cell or is an innocent bystander genetic mutation and the cell doesn't care. That's one explanation. I tend to think that this is the right, at least one, of the factors influence that, tumor spectrum is also because when we first generated mice, knockout mice, that carry only one allele mutated allele of the NF2 gene. So genetically, they're identical to NF2 patient. So we followed the mice, for tumor development, close to 2 years. Turns out that mice do not develop schwannomas, do not develop meningiomas, no ependymomas, but they develop osteosarcoma, fibrosarcoma, liver cancer, highly malignant tumors. So there is a clear, not only tissue specificity of tissue vulnerability to biallelic NF2 loss, but this vulnerability is not conserved across species. No one is working on this, unfortunately. And this is not just specific to NF2. If you look at most tumor suppressor genes, retinoblastoma, a rare tumor of the retina, and that's it. APC, colon tumors, Fonny-Poll-Lindau, very restricted, or tuberosclerosis, all have extremely narrow tumor spectrum. So we already mentioned the fact that study suggests that other genes or epigenetic modifications could be involved and could explain different behaviors of these tumors, because that's something we should keep in mind. These tumors in NF2 patients tends to be multiple. So patients have bilateral vestibular schwannomas, so schwannomas of the acoustic nerve, which is like 95% of the patients. But they can also have schwannomas of other cranial nerve, spinal nerves. And what we see is that, well, first, an important clinical observation that as soon as we make a diagnosis of NF2, and this could be any time between, I think I've seen a 3-year-old NF2 patient, but I also had an 85-year-old diagnosis of NF2. As soon as we do the first MRI, we analyze the tumor burden, saying, okay, this patient has bilateral vestibular schwannoma, trigeminal schwannoma, lower cranial nerve schwannoma, hundreds of spinal nerve, spinal root schwannomas, 3 meningiomas. So the tumor burden will not change during time. So no new tumors will pop up new during life. So that explains that these are developmental tumors, so they start very early in development. And what we see during progression is that they simply get larger, but not all of them. So some grow, some don't, some grow and stop, some never grow. So that's what makes the clinical management of NF2 extremely challenging. And because we sort of, the best therapy for NF2 is do nothing. So what we call the wait and follow or wait and see approach, meaning MRI scans every 6 months in the case of growing tumors, of every year in the case of no growing tumors. So, okay, NF2 mutation, probably other gene mutation that can modify the tumors that are initiated by NF2 loss. So those additional alterations could decide whether the tumor grows or stops, for example. And what are the therapeutic implications of those other genes alteration? It means that our targeted therapy must take care also of those mutations, or hitting the pathway that the main pathway that is deregulated by NF2 loss sufficient to have a therapeutic effect. Finally, all this could lead to personalized medicine, of course, individual treatment option. But in this case, it's not just individual as a person is individual tumor therapy, okay? Because again, we assume that, you know, other events influence the growth, the expressivity of the disease as far as individual tumor response to NF2 loss. So maybe in the near future we will, you know, make biopsies of these tumors and do a complete genomic or whatever omic you can think of analysis, trying to identify those molecular targets that eventually can be coupled to existing treatment, to FDA-approved drugs that have been shown, you know, safe and effective for other tumors and could be repurposed for NF2 tumors. That's the hope. And our working model, definitely. I think that's it.
Wow. All right. Are there any questions for Marco from somebody who hasn't asked a question yet? And a lot of people had fantastic questions and really led in good directions.
Marco, we have some data from one patient and a sibling. Do you know of other databases available for NF2? You obviously must. Also, do you know of RNA databases, gene expression databases, which may allow us to look more into the pathways?
Well, I think those two papers that I showed, 2016 and 2017, that's where the data is. The question is how we get it. I don't have it. We requested it.
I'm sorry, so you requested it, but they didn't?
No answer.
So to the extent...
That's quite usual. So we will need to put some pressure. Eventually it will happen.
So to the extent that you can share with us, could you give us clue on, for the current drug that are in the pipeline, what kind of NF2 biology is this trying to tackle?
What kind of?
What kind of intervention is it trying to tackle?
I mean, nothing fancy or out of the box. You know, it's a classic approach of taking drugs that are approved for other indications in oncology and say, oh, maybe NF2 deficient cells have upregulation of the mTOR pathway, so we use an mTOR inhibitor. And then you have different situations. You know, groups that are, you know, stronger in preclinical testing, try to go as far as they can using whatever model, in vitro or in vivo model they have. Other groups are just, okay, you know, we try. But, you know, nothing, I mean, really, yeah, the beginning. Very frustrating.